the PRINCE trial was a trial of 37 patients, and we had a very rich opportunity to do serial biological profiling on these patients to better understand biomarkers of lutetium-based therapy, which to date have been not very well defined, and we're learning more at an ever-increasing pace as lutetium becomes standard of care in the arsenal of therapies that we have for castration-resistant prostate cancer. So what we did was essentially perform serial PET imaging throughout the trial and serial tumour collections, as well as blood collections for numerous assays, including CTC profiling. So CTC profiling essentially involved collection of a liquid biopsy and enumerating tumour cells, and in particular PSMA positive tumour cells, which is the particular subset of circulating tumour cells we're interested in, to see if tracking those may help us understand responses to treatment, but also parallel what we see clinically in terms of PSA responses, radiographic responses, and symptom responses as well. So, the study was designed such that we would profile circulating tumour cells at numerous time points, and essentially what we found was that a high proportion of patients with castration-resistant prostate cancer have detectable circulating tumour cells, up to 90% or so, and a significant proportion of those circulating tumour cells are PSMA expressing. Between 60 to 70% are PSMA expressing. What we found was that after 12 weeks of treatment with the serial testing, that proportion of PSMA positive CTCs significantly declined under treatment pressure, and the total CTC counts also declined as well. This was also in parallel to declines in PSA and declines in PET metrics, namely tumour load or tumour volume and lesional activity, which is a kind of a mixture of an SUV plus volume. What we found was that by profiling serially, those patients that cleared PSMA positive CTCs seem to have a better clinical outcomes from baseline, in particular improved PSA progression-free survival and improved radiographic progression-free survival, meaning that potentially serial profiling for clearance or non-clearance of PSMA-positive CTCs may be a valuable prognostic biomarker in this space. We're working really hard to understand more about predictive biomarkers for us to be able to understand who on treatment does or does not benefit from ongoing treatment, and that's a work in progress as we do, perform independent validation of this work and increase sample size and so forth.